Welcome to Life and Biology. I'm Dr. Joel Graff, and this video is going to be, excuse me, talking through the slides of a lecture I gave on February 16th, 2022. We're going to talk about mitochondria, some other organelles, and also the cytoskeleton. So we had finished up the previous lecture drawing out mitochondria and also having a short discussion about where the different phases of cellular respiration occur, and with the exception of glycolysis, everything happened in the mitochondria. So in this slide, before we move away from mitochondria, I just wanted to mention that mitochondria have their own genomes, and that's because they were originally a bacteria that was eaten by an archaea way back in the day. Maybe we'll say maybe about 2-3 billion years ago, I think. I shouldn't guess. Let's see. So anyway, the fact that they have genomes is because they originally were a bacteria with its own genome. Now, your mitochondria that you have in your body came from your mom's egg, and you can trace your maternal ancestry by sequencing your mitochondrial genomes. Okay, and I had made the point in class that it used to be that people would get information about their Y chromosome or about their mitochondria, and it was a way of determining some ancestry. The mitochondria being your mom's all the way back, and then the Y chromosome obviously would be your dad's Y chromosome, assuming you have a Y chromosome. Anyway, moving on to talking point number 2. The genome looks like a group of bacteria. And in fact, when we compare its sequence to the known bacteria today, we believe it was a member of the SAR11 group or cluster of bacteria. Clade, I don't know. But anyway, so just by looking at its sequence, we can kind of have a good guess of where it came from. And finally, the genome of the mitochondria has mostly been lost. As a consequence, these bacteria started out as free-living bacteria, but they could never be free-living again. Okay. And in class, I went off for a while on the idea that you might think that pathogens that live in your body would require more genomic information to be able to combat the immune system and sneak around the immune system. But although that is a challenge for pathogens, there's far more different types of challenges out in the typical environment, including temperature changes, humidity changes, all sorts of running out of food. Whereas in your body, you hold a constant temperature and you have a steady supply of food for the bacteria or whatever pathogen you're talking about, making use of your body. So anyway, spent some time talking about that. Moving on, we have chloroplast next, and again, this is a type of organelle in plant cells that is a group of organelles called plastids. And as long as I just talked about the genome of mitochondria, I'll point out that this has its own genome. So these chloroplasts, these organelles within plant cells, originally started out as photosynthetic bacteria. And so when the photosynthetic bacteria was eaten by a eukaryotic cell, and then it decided not to digest it and make use of the fact that it could make glucose, we kept it, and so that's what the plants are. Now, going through the structure, we have the outer mitochondria of the chloroplast and the inner mitochondria of the chloroplast, and those are both smooth, and it's basically just like a double lipid membrane. And that contrasts with the mitochondria, which had all the inner membrane of the mitochondria had all these extended folds and invaginations and stuff throughout. So because they don't have the inner membrane of the chloroplast doing all of those folds and everything, in the chloroplasts, we've got one more set of membranes inside, and these membranes, I drew it out with kind of nice connections between each stack of the different thylakoids. Each layer is called a thylakoid. Then I drew kind of a more shorthand version where I just was connecting them like that to show that the thylakoid stacks were connected. And then this was my third way of drawing it. And this is kind of the laziest way, and it mostly just looks like a stack of pancakes at that point. But anyway, when you get a stack of thylakoids, it's called a grana or granum. And if you've got multiple, they're called grana. So plural is gran, like data, versus granum and datum. Anyway, these have all this extra membrane, as you can see by all the zigs and zags when we're drawing it. And so this is going to be, the extra membrane is going to be the site of photosynthesis, where the electron transport chain and ATP synthesis are located. So that's going to be in the light reactions, which we're going to talk about in a few days when we get into photosynthesis more. But anyway, so that's the chloroplast in a nutshell. One thing I forgot to label on here was stroma, and that's the fluid within a chloroplast. Okay, moving on. Cytoskeleton. We've got 3 different types of cytoskeleton, 3 categories of cytoskeleton: microfilaments, intermediate filaments, and microtubules. The first and third start with the word micro, so it can be a little confusing which is which, but just know that as you go from 1 through 3, the thickness increases. And microfilament, a filament might make you think of a thin wire, or just a very thin fiber, whereas microtubules should make you think about tubes. And so you could imagine a microtubule will have a bigger diameter than a microfilament. Now microfilaments, they're pretty abundant in the cell, and they're made of 2 strands of actin protein polymers that are twisted into a double helix. And so that's a microfilament. So the double helix does not look exactly like the DNA double-stranded, or the DNA double helix, and that's because on a DNA there is unequal gaps between the strands for the minor groove and the major groove. Microfilaments are more or less just a nice consistent twisting of 2 fibers of actin polymers. Okay, intermediate filaments. We've got 2 types to talk about. So these are polymers again. All of these are polymers. And the first type to talk about is keratin. The keratin can be found in skin cells called keratinocytes, and keratinocytes, as they replicate under your skin, they are going to form layers of keratinocytes, and the layers will start dying the closer they are to the surface of your skin. And so then as they die, most of the cell goes away, but you are left with a bunch of this keratin. And so when you touch your own skin or someone else's skin, you are touching, for the most part, a whole bunch of keratin. And well, there's some other stuff there too, but the main protein is going to be keratin. Now, the other protein filament to talk about is laminin. And this is found in the nuclei of different cells. So microtubules, found in the nuclei of different cells. And I'll draw a picture on the next slide to show where you would find this laminin. And finally, microtubules. These are the tube structures. They have about 10 strands of tubulin proteins. The tubulin proteins are alpha tubulin and beta tubulin. And in class, we watched a video called The Inner Life of a Cell. And I don't think it was on this day. Maybe it's the next day. But anyway, the microtubules are very distinct structures. They're going to head from the center of the cell around the nucleus to the outside of the cell. And instead of just describing it in words, I'll move on to the next slide where I've got the microtubules starting towards the nucleus and ending up at the cytoplasm. One thing I didn't draw, that's these red ones here. One thing I didn't draw quite accurately on this is that microtubules are going to come from centrosomes. They're these little organelle structure things, and if you have a microtubule over on this side of the cell and the centrosomes over on this side of the nucleus, and the centrosomes on the other side of the nucleus, that microtubule would actually extend out from the centrosome and then come over here and then reach to the outside of the cell. But the main point is that these are going to be little highways for carrying stuff or moving stuff from the center of the cell, the perinuclear area, that means near the nucleus, to the plasma membrane of the cell. So you're going to have, in the video that we're going to watch, it shows those as well. Shows these microtubules. It shows some transport vesicles getting carried along the microtubules. Okay, so that's the microtubules. Let's go for the intermediate filaments next. So that would be in this cell. It's not a keratinocyte, so we only have laminin to discuss. And laminin is going to be these blue lines running more or less parallel underneath the surface of the nuclear membrane. And so that's going to be giving the nucleus some structure. If you get mutations in your laminin, that can cause the nucleus of a cell to have a weird shape to it. And finally, the microfilament to talk about is the actin. The actin double helix. And they're going to also, just like the laminin, they are also going to run just under the surface of the cell. An exception to that is if you're doing something like phagocytosis, where, let's say, a bacteria was touching up against the cell could recognize it and had the ability to do phagocytosis. You start getting these cross-linkings and these branching of the actin right in that area, and by making a more sturdy surface here, and then by making extended actins, it'll end up pushing the membrane out away from the surface of the cell, because cells want to just be round because that's the most relaxed state. But if they do need to reach out, it's going to be actin under the plasma membrane doing the pushing, or providing the force at least. And then cell-cell contacts. This is just a quick discussion of how plant cells are connected within tissues versus animal cells. So just outside the plasma membrane, the plant cells have cell walls. Meanwhile, animal cells have extracellular matrix, or ECM for short, since that's kind of a mouthful. Then we've got holes between the cells. Holes between plant cells are called plasmodesmata, versus in animal cells, they're gap junctions. I brought up in class that desmata is a term used to describe the holes in Jesus's hands from being nailed to the cross. So if that helps you remember that this is holes between cells, then good. Okay, if you think it was a weird reference, then just ignore it. Then we have glue between cells. So we've got one cell here, and it's got its plasma membrane in blue. Another cell here is plasma membrane in blue. So this would be the cytoplasm side, and this is the cytoplasm side. Plasma membrane, so in between is the extracellular space. These red parts are those cell walls that we had talked about up here. Meanwhile, the glue that's going to be able to stick the cell walls together is called middle lamella. And so that's that. And over here, animal cells can be held together real tightly using tight junctions, hence the name, and desmosomes. In class, I pointed out that there's a lot of tight junctions between cells, like your intestinal epithelial cells, because you want to create a very strong border between what's inside your intestine and the ability for that stuff inside the intestine to get into your body. So these tight junctions help build up the strength of that. Okay, well, that's all the slides we had for that day. The next slide was blank. So anyway, this has been Life in Biology. I'm Dr. Joel Graff, and like and subscribe or not.